Imaging a treatment that does not just manage symptoms but actually repairs what is damaged inside the body. This is the promise of stem cell therapy. Hi everyone, welcome to today's session. I will be discussing stem cell therapy, the future of regenerative medicine, a field that brings hope to millions of patients. Here is the content I will be covering today. Stem cells are very special because they are like the body's master cells. They can do two important things. First, they can make copies of themselves. This is called self-renewal. Second, they can differentiate into various types of cells, a property known as potency. Because of these unique abilities, stem cell therapy uses them to replace damaged or diseased cells, offering new ways to treat conditions that are otherwise very difficult to cure. Stem cells can be grouped into two ways. By potency, totipotent cells like zygote can make all type of cells, even the placenta. Pluripotent stem cells can turn into any cell within the embryo, and multipotent cells are more limited. For example, blood stem cells that only create blood cells. By origin, embryonic stem cells, which come from very early embryos. Adult or somatic stem cells found in our tissues after the birth, and induced pluripotent stem cells, which are pre-programmed adult cells made to act like embryonic stem cells. iPSCs are major scientific breakthrough. Scientists discovered that by adding special genes called as Yamanaka factors, they can turn ordinary adult cells back into stem cells like a state. Why is this important? Because iPSCs can be used in regenerative medicine to replace damaged tissues and help researchers model diseases like Parkinson's or Alzheimer's in the lab. And it also allows safe testing of new drugs on human-like cells. And best of all, iPSCs avoid the ethical concerns linked to the embryonic stem cells. But one of the coolest uses isn't just putting cells back into the body, it's using them to understanding disease itself. This is called disease modeling. Here's how it works. Researchers take a skin cells from a patient with a genetic disease like Parkinson's. They reprogram it into iPSCs and then guide it to become the exact type of brain cell that the disease affects. Suddenly, they have a living human model of that patient's Parkinson's disease in a lab dish. They can watch the disease progress, figure out what's wrong, what's going wrong, and test potential drug. It is a powerful tool for developing new cures. One amazing example is treating age-related macular degeneration, a leading cause of blindness in older people. Here, how it is worked. Patient skin cells are reprogrammed into iPSCs, corrected if necessary using tools like CRISPR, and then developed into retinal cells. These new cells are placed into the eye to replace damaged ones. Clinical trials in Japan and other countries have already shown promising results, giving real hope to patients who once thought blindness was permanent. A more traditional and widely used method is hematopoietic stem cells transplantation, often used in blood cancers like leukemia. Doctors first destroy the patient's diseased bone marrow with the chemotherapy or radiation, then healthy stem cells are given through the bloodstream. These stem cells travel to the bone marrow and start producing normal, healthy blood cells again. These stem cells can come from bone marrow, blood, or umbilical cord blood. There are three kinds of stem cell transplant. Autologous, which choose the patient's own cell. The benefit is no rejections, but cancer cells might return. Allogeneic, which chooses donor cells, it can fight remaining cancer, but may cause immunity problems like graft versus host diseases. Syngeneic, which chooses cells from an identical twin, this is the perfect match, but very rare. Even though stem cells therapy has great promise, it also has challenges. There are risks of tumors if cells grow uncontrollably. The immune system must reject the cells if they are not perfect match. Scientists also need to control the exact way stem cells change into other cell types. Ultimately, these treatments are expensive and not readily accessible to everyone. Another big concern is fake or unapproved stem cell treatment, which can be very dangerous for patients. The future of stem cell therapy is exciting. With gene editing tools like CRISPR, 3D bioprinting of tissues, and robotic system for cell culture, we are moving close to the personalized treatment for many diseases. In the near future, it may be possible to repair heart, restore memory in Alzheimer's patients, or even grow replacement organs. Thank you so much for joining me today. If you enjoyed this video and want to learn more about biotechnology and medicine, don't forget to like, share, and subscribe. See you next time.